Hello, my name is Jason Linville. This lecture is being recorded for CJ 352 Forensic Science Lab 2. This is part of the mitochondrial DNA lecture. This is the part where we talk about the analysis, specifically the amplification and the sequencing of mitochondrial DNA. All right, so we're going to talk about, because, we talked about amplifying nuclear DNA, SDRs. Okay, you do that with the PCR reaction. Do the same thing here. We start out with the PCR reaction. But remember, with nuclear DNA, we just amplified it. We measured it because the allele was the number of repeats. So we could measure the length of that amplified DNA and get our 13 pairs of numbers. Here, we just get amplified DNA, but we need to do something else to it. We need to sequence that amplified DNA. So it actually goes through another reaction called a sequencing reaction. So we're going to talk a little bit about that sequencing reaction. Here's a tip. It's very similar to an amplification reaction. You just have to change. There's 5 things that go into the tube, and a couple of them are just a little bit different. Okay, we need to describe what happens when what the heck is a ddNTP? Is that, that's a mistake, Dr. Levin, you mean DNTP. No, I mean ddNTP, which is, we're going to be introduced to that later in this lecture, Okay, PCR reaction. We start, like I mentioned before, I didn't specifically say this, but when you do a DNA extraction, you've extracted all the nuclear DNA and the cells bust open, you extract the mitochondrial DNA as well. So if you're doing STR analysis, 13 pairs of numbers amplification, squirt, your DNA goes into that tube. For this mitochondrial reaction, it's the same tube. So when we did our extractions earlier this semester, we have your mitochondrial DNA in a tube. Also, we could squirt, do an amplification of your mitochondrial DNA as well. Okay, you don't need to do a separate extraction. This only occurs at the amplification step. So if you remember, when we set up that amplification sheet, we took a little bit of our DNA, I think 2 microliters. We mixed it with Promega mix that contained our TAC and NTPs, and we mixed it with some primers, a primer mix, right? And DNA, and that was our 4 things. Okay, here we're going to have a very similar PCR reaction, all right? But the main thing is, again, we're not amplifying this area of DNA, the STRs. We're amplifying one region of mitochondrial DNA. So what do you guys think has to change? Let me open this up to the chat box. I said we're going to use the same DNA, so that's not going to change. Do you think we're going to have to change our primers? Are we going to change our polymerase? Are we going to change our DNTPs? If we want to amplify mitochondrial DNA, which of those do we need to change? Okay. And if you read ahead in the bullets, you'll probably find out this information. Yeah, it's going to be primers. Because remember, primers attaches to the DNA. It affects, you order different primers, they will attach to different areas of DNA. Okay. So we're still going to use our DNA. We're still going to use TAC polymerase. We're still going to use DNTPs. We're still going to use primers. We just put in different primers than we did for STRs. So essentially the reaction is, again, very similar. We mentioned with STRs a lot of times, I mean, we made our own custom mix, but a lot of times you'll purchase identifier mix, or excuse me, some commercial kit that will amplify the all 13 loci that you need. For this, if you're amplifying one region, you usually don't even have to order the primers from a company. They can just publish the sequence of these primers, and you can actually, well, I say you don't have to order like a trademarked primer mixture from a company. You can actually just order them, or if you have the instrument that makes primers, you can make them yourself. Okay, so you just sort of type in the sequence that you need and order the forward and reverse primer. Okay, that's it for amplification. It's exact same, except the primers are going to be a little bit different. It's still 4 things going to the tube: DNA, TAC polymerase, DNTPs, primers, forward and reverse. 1 set of primers because we're amplifying one area of mitochondrial DNA. Okay, now cycle sequencing. Here, we had our amplified DNA. What did we do with it? We mixed it with formamized and size standard and loaded it on the instrument, and then recorded our 13 pairs of numbers in that long, boring video last week. For mitochondrial DNA, we can't just throw the amplified DNA into an instrument. Okay, because again, we're trying to measure the sequence of it. So we need to put it through a separate sequencing reaction. That's what this is all about. This is about the sequencing reaction. So I may ask you, excuse me, what are the 5 things that go into a sequencing reaction? The answer is the following. DNA, except it's not our DNA from our DNA extraction. It's now the PCR. It's amplified DNA. Okay, so you can either say PCR product or you can say amplified DNA, but it's still DNA. You see that? It's just a little bit different because it's now our amplified DNA. It's that amplified one region of DNA that we're going to sequence. Okay, we also need DNA polymerase. Okay, this is going to be the Taq polymerase, so you can write DNA polymerase, Taq polymerase. Okay, that's going to be the same. Okay, DNTPs, it's not listed here. Okay. Oh my good, Tabby, why are you so angry? All right, the question is, what are the 5 things that go into a sequencing reaction? Okay, 1 is DNA, except it's amplified DNA. 2 is Taq polymerase, exactly the same. 3 is DNTPs, exactly the same. Okay, here's another different. You're going to put just 1 primer into the tube. Okay, here's a little foreshadowing. Like this shows our PCR product and our 1 primer. You're going to just sequence run the sequencing reaction in one direction. You don't want to run it both forwards and backwards. Okay, we're not going to get into a whole lot of details about that, but just 1 primer. Okay, so we got everything. We got DNA except it's amplified DNA, primers except it's 1 primer, Taq polymerase, DNTP. So what's that magical 5th thing? We're still going to put this into a thermal cycler as well, okay, and run this through some cycles. So everything is very similar, okay. The biggest difference, is that in addition to DNTPs, we are also putting in dideoxy NTPs, okay. So let's look at this a little bit, all right. Now, we don't go into a whole lot of detail about how these DNTPs get added, okay? But what hopefully you sort of remember from the nucleotides that we talked about when we introduced DNA is we have a phosphate sugar backbone. This is the sides of the ladder, okay? And the base out here, which this one happens to be an A because it's dideoxy ATP, okay, is the half rung of the ladder, okay? Sides of the ladder, half rung of the ladder. And when we're adding normal DNTPs, okay, we just stack these, right? These links, these half letters, like right next to each other when we're making the DNA. An A, and then a G, and then a C, okay? Well, I can barely even draw a C. Okay. Now, this is where dideoxy nucleotides are different. Down here, in DNTPs, this is an OH group, which essentially means that it has a little link on the end of it, which when you're adding the next, okay, I'm drawing some Lego-like things here, okay, when you're adding the next letter, a T, okay, it snaps into that end, okay, this is like the OH group, okay. So on regular DNTPs, we can snap those pieces together, okay. Snap, 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 because they have the little hook, the receptor on the 3' end, so we can add letters. The dideoxy ATPs, the way I want you guys to think about that is it has this cut off. So it is not a little C at the end there. It just sort of ends with nothing on it. Okay. So what that means is when some new letter comes in to get added, okay, it can't be attached to the DDNTP. All right. So with the DDNTP, I'm going to ask you about 2 things that happen when a DDNTP gets added. One of those is the reaction stops, the elongation stops. A DDNTP, nothing can get added to it. Okay. All right, the other thing that we need, so we're going to go through this sort of full process, but the 2 things I want you to know about DDNTP are, number 1, it has a different end, okay? So it's going to stop the reaction. And also know that the DDNTPs, they are labeled, okay, with fluorescent dyes. I know it's a lot of fluorescent dyes all over the place, but with STRs, the primer was labeled here. The primer is not labeled, okay? But the DDNTP is labeled. Okay, so let's look to see what happens here. Okay, and this is a lousy slide. So let's sort of ignore this because I want to go on the next slide that better shows it. All right, two things are going to happen. Well, basically, you put this in the thermocycler and thermocycling starts, okay? And if you remember, what happens with thermocycling? Okay, well, you have amplified DNA, which is double strand. So it goes up to 94, the amplified DNA comes apart. You go down to like 55 degrees, the primer, just one, attaches. Everything's the same so far, right? You heat it up to 72, the polymerase attaches. Okay, still the same as the PCR we learned before. The primer starts adding, or excuse me, the polymerase starts adding letters. Okay, G, C, T, T, A, G. All right. But now there's not just DNTPs floating around. There's not just regular letters floating around. There's also these special letters, these dideoxy-NTPs. So there's dideoxy-A's, dideoxy-C's. There's not just normal A's and C's floating around. There's special A's and special C's. Okay. There's a heck of a lot more of the normal A's, C's, G's, and T's. But occasionally there's these special ones. So what will happen is the Taq polymerase attaches to the primer, starts adding these letters, and if it adds a normal A, then elongation continues. If it adds a normal G, elongation continues. It keeps adding those normal letters until it just randomly grabs a special one, okay? And when it grabs a special one, the elongation stops. Termination of the elongation, termination of the product. Okay. All right. So when we put our amplified DNA through this cycle sequencing reaction, okay, during the first time through the reaction, remember, there's millions of copies of these. So I'm showing you 4 possible things that can happen, but there are actually like, this happens millions of times. Okay. So what could happen as you separate the DNA, cool it down, okay, again, we have 1 million copies of our amplified DNA. So when you cool it down, 1 million primers attached to 1 million copies of DNA. Okay. And then you heat it up a little bit more to 72, 1 million polymerases attached to these 1 million copies of amplified DNA with the F15 attached. Okay, so the F15 sort of attaches right here. All right. And then the polymerases, all 1 million of them, start adding letters. This just shows 4 things that can happen. Okay, in this first one here, the polymerase right off the bat grabs a special G, okay, and adds that special G, it stops. Okay, this is not getting any longer. At the end of this cycle, okay, this is what it's going to look like. Okay, for this F15, the polymerase adds a regular G and then keeps adding, oh, the second letter it got is special, a T. So this stops. Okay, this one right here adds a G, normal T, and then that third letter it adds is a special A. It stops. Okay. Now, again, these are sort of normally what happens is it'll add like about 100 normal letters before it grabs a special letter, and that special letter is at position 101, okay? But it stops. So can you guys picture this? Like picture it continuing it. It's now 4 normals and it stops at a G, okay? Then 5 normals and it stops at an A. Then 6 normals and it stops at a T. And one cycle through, you start with 1 million copies of DNA, you wind up with 1 million of these, okay? Some look like this, some look like this, some look like this, okay? All the way through to where you have 899 and the 900th one is a special. Now, in reality, sequencing doesn't work well past 500 base pairs, but we're going to sort of ignore that for now. All right, so since millions of copies are there at the start of the reaction, millions of copies are created at each cycle. Okay, so we run this 25 times. This is not amplification. We're not going from 1 million to 2 million to 4 million to 8 million. After the first cycle, we now have 1 million copies of that special cycle sequence DNA. At the end of the second cycle, now we have 2 million. At the end of the third, now we have 3 million. Like each cycle will make 1 million more copies of that special stuff. Okay. So again, we're not amplifying, we're just generating sequenced product is what it's called, sequencing product, as opposed to DNA. Okay. All right. So at the end of this cycle, what do we wind up with? Well, we wind up with thousands of pieces of DNA that look like this. Okay. We end up with thousands of pieces of DNA that look like this. Thousands of pieces. Because remember, the 1 million copies that we create at each cycle, it's sort of spread out between here and like 500 different types. Okay. You know, this has 1, 2, 3, 4, 5, 6, 7 normal letters and then a special letter. Out here, we could have 499 normal letters, and then the 500th one is special. Okay. So we have that. And notice how now we have and they're all different 1 base pair in length. Okay. This is whatever F15 is. Let's say F15 is 20 base pairs long. This one is 21 base pairs. This is 22, 23, 24, 25, 26. If we separate these in a capillary, what's going to come out first? The smaller one, the 21 base pairs long. What's going to come out next? The 22. So can you guys see how these will line up and migrate if they're traveling this way in a capillary? This one's going to, and this is the detector right here. This is going to hit the detector first, create that little peak. Okay, this is going to hit the detector second, create that little peak. This is going to hit the detector third, fourth, and the sequence up here is the same as the sequence GGTACTTT. It's the same as the sequence here. Okay. So this is, I know it's difficult to visualize, okay, but imagine that you're creating all of these that are all mixed up together, thousands of copies of each type, and then you separate them to line them up in order. So the first letter in the sequence comes out first. Okay. Now, how am I going to ask this on a multiple choice, or excuse me, how am I going to ask this on the quiz or exam? I'm going to ask you, what 2 things happen when a ddNTP is added to the sequencing reaction? Okay. And those answers are, number 1, the elongation stops. And number 2, also notice here, the piece of DNA is labeled with a fluorescent dye, but be more specific than that. The piece of DNA is labeled with a dye that corresponds to the last letter. Notice here that all G's in our mixture of ddNTPs, all G's are labeled with a yellow dye. All T's are labeled with a red dye. A's are labeled with a green dye, and C's are labeled with a blue dye. All right. And I think I need to double check all my colors here. All right. But the thing is, since those ddNTPs, it's a mixture of those special letters that will stop the elongation. And also all the T's are one color, all the G's are another color, all the C's are another color, all the A's are another color. You can separate them, line them up, and that gives you your sequence. Okay. So for mitochondrial DNA, we're going to just write down this sequence, and it's going to be 900 base pairs long. Now, like I said, in reality, it's tough to do. So you usually have to sequence one section of it and then stop and then sequence another section of it. But you guys, if you visualize it as 900, that's going to be fine. Okay. This slide you do not need to know, but wait a minute. After we run that cycle sequencing reaction, do we just throw it on the instrument? Well, yeah, pretty much, but we prepare it. We need to get it in formamide to get it in single-stranded form. But notice here, every base is going to be 1 base pair apart. We care about the order. We don't care about the size. We don't add a size standard in here, okay? You just have to mix your sequencing product with formamide and then run it, okay? And these differences are more important for the graduate class, but the polymer is a little bit tighter to create better separation, and you run it for a little bit longer through the capillary to get better separation, but that's it. Okay. All right, I'm going to go ahead and stop this mini recording, and and then we will continue on with the